Our first presentation is by Tuba Harris from Baylor University. And you may share your screen, and when you're beginning, you may begin when you're ready.
Okay. Hi, everyone. Today I will be talking about my research in analyzing gene regulatory enhancers in hepatocellular carcinoma. So the main objectives for our research are to find evidence of redundant enhancers and to quantify the similarity between different enhancer sequences, as well as understanding effects of regulatory genetic variation in liver cells. So to start off with, what are redundant enhancers, or what are enhancers in general? So enhancers are gene regulatory elements that can help with the growth and development of cells and tissues. They also contain binding sites for transcription factors. So they help with activating transcription of genes, and mutations in these regions can cause an appearance of traits that were not previously present, and they can participate in disease processes. So this image you'll see right here, that is preaxial polydactyly. That is a polydactyly where an additional digit grows towards the first digit of the hand or the fifth. And that is actually caused by a point mutation in the ZRS enhancer. So to start off with, this is kind of what an enhancer looks like. So enhancers are typically far away from the gene that they're associated with. So as we can see in part A of this image, and then they're brought into closer proximity as seen from part B and C through chromatin looping. That allows it to activate transcription and for enhancers to work with gene expression. So in our research specifically, we are looking at combinations of enhancers because we have found that combinations of enhancers that regulate the same gene have redundant elements. This image right here is from a study done in 2018, and it was my study. So if you can see like the second image right there, that is showing redundant enhancers in full effect, and you can see it's causing the growth of additional digits, and not just one, but multiple. And then if you see the very last image on the end, that is also showing growth of an additional digit. So again, that is telling us that redundant enhancers are playing a role in disease processes. So for the purpose of our research, we are focusing our research on hepatocellular carcinoma, or HCC. HCC is the most common type of liver cancer, and it often has a terminal prognosis. Approximately 25% of all HCC cases appear with genetic mutations. So for our research, we are focusing on 4 genes that are commonly associated with HCC. So first we have TP53. TP53 helps control the cell cycle, and a mutation in this gene can cause abnormal cell growth, and it can result in cancer. We also have CCND1. An amplification of this specific gene can lead to overexpression in the activation of oncogenic signaling pathways. And then we have CTNNB1. An increase in the WNT beta catenin pathway is associated with a mutation in this gene, and that can lead to heightened oncogenic activity. And then lastly, we have IGF2R, which is a tumor suppressor gene, and a mutation in that specific gene can result in the loss of heterozygosity. So in our research, we started with getting enhancer sequences, running them through FEMA, which is an algorithm, and then obtaining information from there. And then after getting our information, we ran multiple tests and multiple analyses on our data. So first off, I'm going to start with getting our data. So in our research, we are analyzing over 70,000 human enhancer sequences in liver cells with predicted gene targets to find evidence of redundant enhancers. So to get these sequences, we downloaded H3K27AC, and H3K4 ME3 ChIP-seq peaks. And in these peaks, we were essentially looking at the H3K27AC peaks, and we labeled them as enhancers. And to use them in our data, we made sure that these peaks overlapped by less than 50% with the peaks that were present from the H3K4 ME3 peaks. Once we got these enhancer sequences, we ran them through an algorithm called FIMO. FIMO basically finds motifs that were present in these sequences, and it also treats each of these motifs independently. So after getting that, we created like a really large data frame for our data. So our data frame essentially was for the entire human genome, and it gave us all the information for our enhancer sequences. It told us what chromosome it was associated with, as well as the start and stop coordinates, the gene it was associated with, and then most importantly, it also told us if that motif was present in our sequence. So like you can see a lot of zeros in our data frame. So a zero basically told us that that specific motif was not present in our that motif was not present in that specific enhancer sequence. But you'll see right here we have a 2. So that 2 essentially tells us that not only is that motif present in that enhancer sequence, but it also tells us that there are 2 of those motifs present. So after getting this information, we wanted to look at what were the most prevalent motifs present in the human genome. And we found that MAC was the most prevalent. It had over 2 million hits. Then we had VEGF1 and ZN467, each of which had over 1.7 million hits. So MAC essentially participates in transcription termination and polyadenation, and deregulated expression of MAC is correlated with various tissue malignancies. And then we also have VEGF1 that is involved in normal and abnormal cell proliferation and differentiation. And then lastly, we have ZN467, and that specific motif is a transcription factor for several genes, and it can help recruit histone deacetylase complexes. So essentially that also participates in transcription termination. So after getting all of this information, in our study, we are studying like a combination of enhancers because we know that they have redundant elements, and if they have redundant elements, they can participate in, they can affect gene expression and participate in disease processes. So for the purpose of our research, specifically HCC. So first off, we had our entire data set. We reduced our data set into 4 smaller data sets, 1 data set for each of the genes we were looking at for HCC. After breaking down our data set, we ran a principal component analysis, or a PCA. The PCA essentially is used to reduce the number of variables in a data set while preserving as much information as possible. So for our analysis, we were looking for clustering patterns because those clusters would tell us that if redundant enhancers are playing a role. So after we further broke our data down, we plotted our PCAs for all 4 of our genes, but then we also layered a kernel density plot on top of our PCAs. So for all 4 of the genes, you'll see that they have similar clustering patterns. They're all clustered close to 0 for PC1 and PC2. And that essentially tells us that there's not much variation in our transcription factor binding profiles across the enhancer sequences linked to the same gene target. And that again indicates that they may demonstrate similar function. So the kernel density plots, they show us that the contours highlight like density in or highlight density in those particular regions. And again, that is giving us evidence of redundant enhancers, which is what we were looking at. So to further analyze these clusters in those areas, we introduced tissue specificity scores because we wanted to see what are setting those clusters together and why are outliers in our data not part of those clusters. So we developed a scoring system called tissue specificity scores. Tissue specificity scores range from 0 to 1, 0 showing broad activity, so and then 1 showing narrow activity. So a score of 1, like you can see in this fourth column or fourth row, that would essentially say that that specific enhancer sequence is only active in liver tissues, but then a score of 0 would indicate that that enhancer sequence is active across 10 different tissues. So with that, we plotted all of our tissue specificity scores for each of our genes that we were looking at. And our scores were really variable across all of the HCC related genes. But for IGF2R, that was our only gene that had really skewed results. It goes really skewed to the right, indicating that the tissue, that it was the most the most of the enhancer sequences for that specific gene were only expressed in liver tissues. But for the rest of the genes, it was really a variable across the board. So for IGF2R, that may be because it is a tumor suppressor gene, and the rest of the genes really are associated with cell cycle or just like oncogenic pathways. But that one is just specifically a tumor suppressor gene, which may show why we're getting this data. And then overall, we are in our research, we used a pipeline method to determine problem motifs for each gene. We found that MAZ, VEZF1, and ZN467 are all related to termination of transcription and cell proliferation. And then we also found from our analysis that clustered regions in gene candidates are candidates for redundant enhancers, and they have similar transcription factor binding profiles. And then lastly, we found that tissue specificity scores quantify like the number of tissues in which these enhancer sequences appear active, and IFG2R was really the only one that had really skewed results. So for future directions, we want to determine specific transcription factors that set apart the outliers in our PCAs. And then we also know that redundant enhancers and their activities facilitate the understanding of gene regulatory networks, and they are influenced in controlling disease-associated genes. And then furthermore, we want to develop another scoring system to quantify how redundant a group of enhancers are using PCA and other dimensionality reduction techniques. Thank you.
Thank you, Tuba. So does the audience have any questions for Tuba regarding her presentation? No? All right, judges, do you have any questions for Tuba regarding her presentation?
Hello, Tuba. Excellent work, excellent job. Also, I'd like to just go ahead and take this moment to encourage all of the other presenters to ask questions of each other's talks and engage with each other. And that's one of the things I'll be looking for from you guys. My question to you is a very general one. And the question is, what kind of benefit might there be to having redundant enhancers from an evolutionary perspective?
Yes, so redundant enhancers essentially from an evolutionary perspective, as we are aging, there are certain parts, certain characteristics that we're not using from previous years. So those redundant enhancers may be participating in that, but finding redundant enhancers now can help control disease processes to like stop the progression of certain diseases if we catch them before. Like for our study specifically, if we can find evidence of this, and if we can look at people who are already predisposed to liver cancer or HCC, we may be able to like stop it from developing further and catching it at an earlier stage. So those are just some of the reasons for our research.
Okay, I'll give the judges a few
Excellent. Thank you, Tuba.
Thank you.
All right. Do the judges need any more time to ask any more questions or write any more notes or writing feedback before we begin our next presentation?
I'm ready.
I'm okay.
All right, sounds good. So our next presentation is by Sinchana Basur from Baylor University, and you may begin whenever you're ready and share your screen. Thank you.
Sorry about that. Give me 1 second. Can you guys see the screen okay? Okay, sounds great. Let me minimize. A lot of the Zoom things are just everywhere on my screen right now. Okay. Well, hello everyone. My name is Sinchana Basur, and today I will be sharing with you how integrin G and CK2 may mediate GIST-induced sodium reabsorption through epithelial sodium channel, or ENaC. And this is research that I conducted over the past summer at the University of Texas Health Science Center in San Antonio. So hypertension is an increasing predicament that is facing the world's population, with over 1 billion people who are affected, but only 1 in 5 actually have the condition under control. Nearly half of the adults in the United States alone are affected, and it's costing us a heavy sum of around $131 billion every year. So to address this growing issue, the epithelial sodium channel may be a potential candidate for medical therapeutics. And just a little bit about the epithelial sodium channel, or ENaC. It is specific to the apical membrane of principal cells, which are located in the distal tubule and collecting duct. The primary function of ENaC is to reabsorb sodium and from urine in order to increase blood pressure, and therefore regulation of ENaC is very critical to maintaining the body's blood pressure, and that makes it a great target for several of our body's hormones, including vasopressin. And vasopressin is a hormone that is produced by the posterior pituitary gland in response to low blood pressure. Vasopressin acts by binding to its corresponding G protein coupled receptor on principal cells called B2. B2 is a GS-GPCR, which is stimulatory, and so it causes downstream activation of cyclic AMP and then PKA. And PKA is then able to activate a number of other pathways, one of which may affect ENaC. And GS-DREAD, just a little bit about it, it's an artificial receptor, and essentially it allows us to study this pathway, and we're testing it to make sure that it can represent the V2-based knock-in model that we were hoping to use, which is conditionally expressed in principal cells and only activated when exposed to this molecule called clozapine N oxide, or CNO. So to start off this summer, we hope to evaluate whether GS-DREAD is a reasonable model for the V2 receptor in our case, and then use it to determine how ENaC is affected. We believe that GS-DREAD, when activated in principal cells, would result in a signaling pathway that increases ENaC activity and thereby increasing sodium reabsorption. So to test this hypothesis, we used immunofluorescence microscopy on kidney sections from GS-DREAD mice. And ENaC was visualized in the red channel, and GS-DREAD was seen in the green channel using its intrinsic GFP tag. So in the wild type, you can see that there is no green fluorescence, which is good because there shouldn't be any GS-DREAD there. And then in figure B, you can see that there is co-localization of the red and that indicates that the GS-DREAD receptor and ENaC are co-expressed in principal cells. So we were good to move on. And here we conducted cell-attached patch clamping. to study the channel activity of ENaC. And we isolated and split open tubules from GS-DREAD and wild-type mice to expose the apical membrane of the principal cells, which is where ENaC is located. And we compared untreated groups with CNO-treated tubules. And in figure A, you can see that the wild-type tubules have reasonably similar ENaC activity as the CNO treatment. And in figure B, you can see that the GS-DREAD tubules had much more robust ENaC activity when they were treated with CNO versus just no treatment. So in this slide, we're just kind of summarizing what we saw in the previous slide, but the biggest takeaway that I want you guys to have is from figure E. And what you can see in this figure that you couldn't see in the previous slide was that not only did CNO treatment increase ENaC activity, it also increased the number of channels that opened in response to the GS-DREAD pathway being activated. Okay, and then we also did fluorescence recovery after photobleaching or FRAP studies on COS7 cells, which are monkey kidney cells. And we transfected them with the complete ENaC protein tagged with YFP or yellow fluorescence protein. And photobleaching basically causes the membrane proteins to retract into the cytosol. And then if you give it time to recover, the membrane proteins return to the membrane. We use turf microscopy, which allows us to examine the proteins specifically at the membrane level. And we compared the fluorescence recovery between untreated and cyclic AMP analog treated cells. We observed that there was an increased fluorescence recovery in the cyclic AMP treated cells compared to the untreated cells. And this indicates that cyclic AMP increases ENaC translocation to the plasma membrane. And this is basically summarized in figures B and C. We also wanted to confirm that the electrical activity of the in vitro model is consistent with what we observe in the tubules from the previous slides. And figure D and E, basically we confirm the patch clamp data seen before. So far, we have evidence that the GS-DREAD receptor co-expresses with ENaC, which makes it a good model for the GS-based V2 receptor. And the FRAP and patch clamp experiments indicated that the activation of GS signaling pathway increases ENaC activity, but we found it was very interesting to me that the CNO treatment led to a greater number of ENaC present in the plasma membrane, and that treatment with cyclic AMP led to an increase in ENaC translocation to the plasma membrane. So overall, there were more ENaC units at the plasma membrane. And there seemed to be a connection here, but we didn't know the particular proteins involved or the mechanism behind it, so we decided to continue exploring. And one hypothesis I had was that ankyrin G and casein kinase 2 were involved. Ankyrin G or ANKG is a cytoskeletal protein that works with kinesin and anterograde transport toward the plasma membrane. And CK2 or casein kinase 2 is a protein kinase that is specific to serine and threonine residues. And we speculated that the GS signaling pathway ultimately led to enhanced binding of ankyrin G to CK2 phosphorylated ENaC, which would mediate ENaC translocation to the plasma membrane through through kinesin. Okay, and so ENaC has 3 subunits, alpha, beta, and gamma. The C-terminus of the ENaC's beta subunit is particularly important for its intracellular regulation. And if we look at its primary sequence of the C-terminus, we can see that there is a CK2 phosphorylation site that actually overlaps with the ankyrin G binding site. And to test our hypothesis that CK2 phosphorylation mediates ankyrin G binding to ENaC, we mutated the highlighted serine on the beta C subunit to an alanine, which prevents phosphorylation at the site. We then conducted FRET or Forster resonance energy transfer experiments with CFP tagged ankyrin G, which is cyan or bluish color, and a YFP tagged beta ENaC, which is yellow. And FRET works by measuring the amount of fluorescence that is transferred because the two proteins are very, very close together. And it's, I believe, a maximum of 8 to 10 nanometers. So very, very close. And so in figure A, we can see that the cells transfected with the wild type beta subunit C terminus, those cells were treated with TBB, a CK2 inhibitor, and they exhibited a lower fluorescence transfer. But when we treated the wild type cells with CK2, the transfer was profoundly greater. And it may be a little easier to see these results summarized in figure B, which essentially quantifies the fluorescence transfer. And in figure C, you can see that the experiments conducted with the mutated form of beta C and exposed to the same treatments, there was no difference in the fluorescence transfer across the groups. And so this indicated to us that CK2 phosphorylation was integral to the translocation of ENaC to the plasma membrane and also the interaction of ENaC with INK-G. And that is something that we wanted to continue exploring. And so we started to conduct binding interactions with ENaC and INK-G using a synthesized peptide of the ENaC beta C peptide. And we then solubilized and performed binding assays with this. So we attempted to phosphorylate the peptide initially, but the reaction caused the beta C peptide to crash out of solution. And we attempted to repeat this experiment, but because it kept crashing out, we just proceeded to conduct the isothermal calorimetry on the unphosphorylated peptide. And with this, we titrated INK-G with the beta C peptide and observed the binding pattern. And ITC, or isothermal calorimetry, works by detecting the change in heat, which is an indicator of whether a reaction is exothermic and occurs spontaneously. And we can then calculate the binding affinity of the two molecules using this data. And if we compare the resulting curve with the control assay, it was apparent that we didn't detect any binding between INK-G and the unphosphorylated beta C. But this was expected, again, because we hypothesized that a phosphorylated beta C is necessary for INK-G binding. So our future steps would be to repeat this experiment with the phosphorylated beta C. So to conclude, the GS pathway leads to increased ENaC activity, which is partially due to increased trafficking to the plasma membrane. And I think further research would be crucial to determine whether GS signaling ultimately leads to coordination between CK2 and INK-G to transport ENaC to the plasma membrane. Thank you so much for listening to my presentation, and I will take questions at this point.
Do any other presenters or judges have any questions for Sinchana?
I have a question. What led you to look at CK2 and INK-G? Like, what made you suspect there might be a connection there?
Yeah, that's a great question. So again, I mentioned that the intracellular domains are sections of ENaC were something that we looked at because we knew that there was some coordination between proteins there. And we've found there's previous research in this lab that has shown a lot of interaction with other proteins. And so I used some software online just to look at any like phosphorylation sites, anything that might lead us to believe there is interaction with other proteins. And I noticed that there was a pretty high hit with CK2 in this region. And we had already been studying INK-G a little bit, and I noticed that it was overlapping. And that was when I was like, okay, maybe there's a connection here. So I did some literature search into INK-G and also casein kinase 2 independently, but I saw that there wasn't any research looking at the two of them working in conjunction. And so that was whenever this hypothesis kind of formed. Yeah, does that answer your question?
Yeah, thank you.
I have a quick question. So the V2 receptor, it's like it's a GPCR that like activates protein kinase A, and like we know that like protein kinase A is involved with a ton of other pathways within the cell. So were you guys concerned at all with like how like it could impact those other pathways aside from activating ENaC?
Yeah, that's a great question. There's a lot of crosstalk in cells, but I don't know how else protein kinase A could have influenced um, this particular pathway. I'm not sure how it affect ENaC. Or are you talking about like other aspects of the cell and other pathways that might be relevant to, I guess, protein, or sorry, with ion channel like activity? Is that what you're asking?
Oh, I was just asking about like so like by going through that
Mhm.
signaling cascade pathway, the end result does it does bring ENaC to the cell surface, correct?
That is what we are hypothesizing, yes.
Okay. And then but through that, it does seem to follow a similar pathway that like some other cell processes
Mhm.
also use. One of those being through protein kinase A, which was part of the pathway. So I was just
Mhm.
curious, like since that is so involved with a ton of other signaling cascades, a lot of them being like transcription factor
Yes,
activation
yes.
and stuff,
Mhm.
like what concerns y'all had about like how that could impact the rest of the cell.
Yeah, so when we designed the experiment, we made sure to use an agonist and compared it to one without it. And so I mentioned the GS-DREAD, that's an artificial receptor. And so we're inserting that into these mice. And so that way we can make sure that compared to the baseline, the transgenic mice, that there is a difference in ENaC activity because of this pathway, because we inserted this pathway. And so if there is other stuff going on, because we compared the two different mice, we can kind of say that what we observed with ENaC is mainly due to the activation of this direct pathway.
Okay, awesome. That answers my question. Thank you.
I have a question.
Mhm.
So you mentioned that you would repeat the experiment with phosphorylated beta C. Other than that, like, would you repeat the experiment like multiple trials, or like, would you do anything else other than that to like confirm your hypothesis?
Mhm. And so that would just be the first step. And then, of course, we also believed that anchoring G and CK2 are involved in terms of like binding of CK2 to the peptide initially to phosphorylate it. And so we would need to see if that actually occurs. And that's why we needed the phosphorylated beta C peptide. So I did want to observe if that happened spontaneously or if that was something that didn't exactly occur. And so I wanted to do a little bit more thorough search of that through maybe not ITC, but just like phosphorylation studies. And then again, proceed with ITC with the phosphorylated peptide. And then at that point, again, anchoring G and the beta C. And you could keep going because there's many aspects to this pathway. So anchoring G works with kinesin. And so I'd want to see whether or not once anchoring G has bound to the beta C, if it does bind to the beta C peptide, whether that would then work with kinesin, which we believe would allow it to be transported to the plasma membrane. So yeah, that would be one aspect that I would definitely look into.
Okay.
All right, so we're running a little behind schedule. And so do the judges have any other questions before we go ahead and move on to the next presenter?
Yes, I'd just like to ask one quick question, Sinchana. Great work. So could you just outline for me which parts of these this data that you were generating data for? You showed a lot of different kinds of experiments. And just for you and maybe for the following presenters to help clarify for us, what was it that you directly produced?
Yes, absolutely. So I'm still sharing my screen. I can go back. I performed the immunofluorescence. And I also did a little bit of patch clamping, but also with some help of other lab members, because that is a very, very complex technique. And so I did get a couple of the wild type experiments, but it required a lot of help. And then I also completed the, the FRAP experiment. And then I was able to work with another lab member to complete the FRET. I actually wasn't allowed to use some of the equipment, so I had to work with them. And then the ITC I also did. And I also tried to synthesize the beta-C peptide myself. And we ran into a lot of problems because it is a membrane protein and we were just getting a very small portion of the peptide, of the protein. And so that was very unstable. And instead we purchased a peptide that was synthesized by a company.
Great, thank you so much.
All right, our next presenter is Priyanka Senthil, and whenever you're ready, you can begin. I will stop the recording. You may go ahead whenever you're ready to start.
All right. Hello, everyone. Thank you for staying. Even though we're running a little bit behind, we have a bit of a different presentation for you today. This is a methodology poster, which we did on our model constructing and optimizing these mini bioreactor chambers to study the gut microbiota and its different community dynamics in a system that more closely mimics the human gut. So. All right, a little bit of background. This central picture you see is the kind of existing, more commonplace large-scale bioreactor model. You can see how large those are. They are commonly used in some labs, but also a lot of food industries, and they aren't as easy to control for small controlled laboratory experiments, such as what we are interested in focusing on. So on the right, you can see a piece of our model, which is significantly smaller, which makes it a lot easier to control nutrient flow and other variables such as waste, pH, so that we can better control stationary phase of these bacterial communities and study more specific interactions and dynamics within that.
And one of the key benefits to these like continuous flow culture models is that we're able to remove the waste and add media, which allows for extended growth times. Because if you were to leave this alone without any sort of continuous flow model, the bacteria will follow a general curve and then die off. And so this is really helpful so that we can more accurately simulate how things are going on in the human body. And it also allows for a more high throughput model because we can put more fluids through, and we can also kind of like test more things rather than having to, if with these large scale models, if we were to use something like that, it would take a lot more media. We would have to wait longer times. So our model is generally like faster to use, even though it can still be used for, we've got it up to around 2 weeks.
So our major goals with this project were to first maintain a single bacterial culture within our MBRA system and see if we can maintain it in stationary phase, look at the characteristics within that, and then eventually move on to the reanimation of stool samples so that we can see multiple different species living in one community, which more closely mimics what is happening in your gut, because you aren't the gut isn't colonized by only one community, by only one species.
So what led us to building this model that we have currently? There are current existing models. and we'll go over two of the ones that we were in contact with labs that were using them. I actually traveled to Baylor College of Medicine in Houston last summer to learn more about this model that we're looking at right here. And we intentionally, the first intention was for us to like gather the supplies, learn more about the system, and then completely replicate it at Baylor in Waco. But we did come to a couple issues, and with also the communication that we had with another model over at Mayo Clinic, we decided to kind of combine things that we liked from each of them in a way that best serves our purposes at Baylor University with what we have available. So the main difference between the two, I'll go over now. So the benefits to the Baylor College of Medicine system, these chambers that we have right here are 3D printed. And so they were able to like have a dedicated sample port, which would allow for like easier use whenever you want to like inject or take out media and you want to like analyze whatever's going on in there, see how the bacteria are growing. And so what they would often do is they would take like daily or even like 2 times a day samples and then plot those differences over like 2 weeks and see what's changing in the bacterial levels in response to whatever they want to use. It could be a drug, it could be different types of media. They even test like the effects of like a keto diet or something on human fecal microbiota samples. So, but then what they actually are best for at Baylor College of Medicine, they actually work with C. difficile. And so they're able to use that because they have this convenient waste management system. And it basically replaces water with media in like a dedicated chamber that isn't exposed to the laboratory air. And so I really liked that method because as we'll see in the next one, it's a little bit safer to use with like disease causing bacteria. And then another nice thing that they had was they used 2 pumps on either side. And so by doing that, they allowed the pressure inside the chambers with the pumps to bring the media in from the bottles out to the waste without any sort of issues. You know, if you just have one pump, it can cause overflow and stuff. And so by having 2 separate pumps, it really helped minimize the issues that could have happened with that. They also, if you can see here, they put little stir bars in each of them and that fit over this giant stir plate. And by putting it on there, you can turn on the stir bars and then that allows you to mix the media. And so I liked that method, but we actually ended up changing it around a little bit because the 3D printed chambers, we noticed that a lot of them do get like cracked or like they aren't the best to go into an autoclave. And that was their protocol, was to put it into an autoclave and that sterilized it. But the problem with having like 6 chambers all in the same block is if you lose one, you know, due to contamination, or if one of them just pops open, that does tend to happen with these threads. After putting in the autoclave, they can just come undone and you just lose that replicate. And so it's important to, you know, try to keep all of your replicates. Here they have 24 replicates. We are able to do 6 currently, but we can expand our model as needed to allow for more, but we don't want to lose any of those, obviously. And so at the Mayo Clinic, I was in contact with them via like FaceTime and Zoom. So it's a little bit less detailed, but their model was a little simpler as far as like the pump and waste management system. They also utilized an IV bag rather than bottled media for their delivery of media. And one key difference that they had was the use of these round bottom Falcon tubes that allowed for like individual chambers to be dealt with as needed rather than having all of them in a row attached to each other. So I really liked that method. The other nice thing is that these Falcon tubes are a way more reliable enclosure. We see much less issues with like cracking and stuff that we had with the 3D printed model. And so we really, after going back and forth between the two, we did try both of them. We actually preferred this model because or just this piece of the model. It allowed for easier management of the media, and by using a three-way stopcock on the outgoing port, we can still sample without having to have a dedicated port, even though it would be nice. But as you can see, we had to use a hypodermic needle stabbed into the stopper. And so there's generally limited space. And so that's why we ended up using the three-way stopcock. The one drawback that they had was the IV bag that they used. Although it is convenient to you can just inject and fill, it is susceptible to contamination more so than the bottled media method, because that can just be autoclaved and then you can screw on your tubing and then you're ready to go. With the IV bag, you have to fill it under a hood, and you also have to screw it on. And that issue right there is that there is a possibility that it can leak, or the media comes out while you're swapping out these connections. And unfortunately, when we're working inside of an anaerobic chamber or even outside, it is more difficult to manipulate this contraption right here. So I think that we in the future are going to not be using the IV bag. So I want to show you guys our model. It does have an IV bag, but we decided that we will be removing that and switching over to bottled media. But everything else is kind of a combination of the two. So we have one pump on either side. We have 6 replicates, but if we want to increase our number of replicates, we can. We have it on a thermo shaker that allows for mixing of the solutions. And you can see the three-way stopcocks that we implemented so that we can still sample and add as needed. And then the waste management system, this is exactly how they had it at Mayo Clinic. And we were only using this one as we are testing single bacteria / any sort of contamination tests. We didn't really need to worry too much about contamination of the rest of the lab. And so if we do choose to move forward with fecal samples or any other more complex or disease-causing bacteria, we will be switching over to that better waste management system, because it's a lot safer for people around the lab. But this right here, we were just doing contamination tests, and then we also grew Pseudomonas aeruginosa. So if we, by looking into the sterilization protocol that we had, we chose not to put it into the autoclave because we wanted to sterilize it using liquids. That way it didn't get damaged. And so for the sake of time, we're going to move on. But basically, the sterilization protocol that we used actually did end up working, because our contamination test did show up as lower OD readings throughout. We were actually maintaining a level of 0 throughout, because it was kind of varying back and forth. We suspect that this could have been issues with the nanodrop. But then we did end up growing Pseudomonas aeruginosa for 2 weeks, and we inoculated on day 7. And so as you can see, the growth curve, it went right up to around 1.1 or 0.1, and it allowed for a continued stationary phase growth for multiple days. It's really wonderful. So future plans, we want to test co-cultures. We hope to put human stool samples into the chambers, and hopefully test dietary effects on indole production, polymicrobial communities. There's a lot of stuff that we can do with this, hopefully. And thank you so much for watching our presentation. And we can open up the floor for questions.
Hi, Quinn and Caitlin. Thank you guys for the presentation. I really liked it. I just had a quick question about contamination. And so I was wondering, how often do you check for contamination? And what do you use to check for contamination? Since I know for cell lines, we use mycoplasma testing. I was wondering if that's the same thing that you guys use or if it's a different method.
Yeah, so
oh, sorry, Caitlin, you can go.
I can go. So every day, pretty much once a day, we would take out, using the three-way stopcock, take out some media and do both OD readings, optical density readings, which is what you saw on the graph, and then also plate streaking. So just streaking a plate and seeing if there's any growth, which we did not see any growth. And then we showed you the the OD readings. So we were pretty confident in our lack of contamination at this point.
Yeah, you can see our control wells. When we did grow Pseudomonas, we did keep 3 control wells, and they all were still at 0 by the end of the study. These were our 3 wells that did have Pseudomonas in them.
Thank you.
Mhm.
We can take one more question from the audience, and then if the judges have any questions, they can go ahead and ask.
Okay, I'll go ahead and ask a question. So what does continuous stationary phase really mean for these bacteria? Do they continue to divide? If so, is it slower during the logarithmic growth phase? Or do they enter somewhat of a quiescent or static phase?
So when they do enter a stationary phase, they do tend to grow slower, which is nice because the removal of the dead cells, as we are going through log phase, we have to worry about the ones that are dying quicker. But once it gets into stationary phase, it starts to become a little bit more regulated. Like you can see that the cells are maintaining the same levels of growth and dying at the same times. And so this allows us to like really look at whatever we want to test, because that is usually how it looks in the human microbiome. We see that kind of same exact pattern. So our goal was to keep the stationary phase because that's how it usually looks in the human gut.
Hi, I have a quick question. So would you try to culture some mixture of the bacteria to see if it's like you can maintain the stationary phase for 2 weeks without contamination?
Yeah, so that's one of our next goals. We have a couple. One, we want to move into the anaerobic chamber that we have and start growing anaerobic bacteria, specifically Fusobacterium nucleatum. And then we also, like I mentioned, we have several stool samples from different healthy and colorectal cancer patients, which is what our lab focuses on. And so our goal would be to take those stool samples and reanimate the entire community that's in there and see if we can maintain that, which would more closely mix what happens in the gut, because in the gut, your microbes aren't living in isolation like we often see in the lab. So that can have a big effect on their metabolite production, their reaction to those, and other properties that we are interested in studying.
That's cool. Thank you.
I have a quick question. So when you're removing the waste, how do you ensure that there's no flow back, or that none of the waste is like flowing back into the chamber?
So the system, it utilizes like a pressure gradient that causes everything to move unidirectionally. And so by having 2 pumps, it's constantly, both of them are turning in the same direction. Everything is going in one direction. There could potentially be flow back of waste if you were to like lose that pressure. So by unclipping any of the lines. So that is something that we try to avoid. And it doesn't usually happen if you don't touch anything and you let it just move slowly. But yeah, through a pressure gradient, everything moves one direction, and it actually sucks the waste off of the each of the chambers, and then it goes directly into the bleach. So yeah, that's the whole goal, is to keep it moving one direction.
Got it. Thank you.
I have one last question, if that's okay.
Sure.
You mentioned the anaerobic chamber. What structural or like functional differences did you have to incorporate for that chamber compared to the other one?
Yeah, so the anaerobic chamber is just a huge kind of bubble structure in our lab that we eventually plan on moving our entire system into, including the media supply, the pumps, the mini chambers, which is what the MBRAs are, and then the waste management system. So there are definitely going to be some difficulties getting that into the chamber through the airlock and everything. And just working in the anaerobic chamber, you have these giant rubber gloves you have to work through. So it does make things more difficult, like Quinn mentioned earlier, especially with the IV bags, which is why we are going to try moving to jars for media supply. So it'll just change the like mechanical logistics of everything quite a bit, which is something we've been trying to keep in mind while building this.
Thank you.
All right, so I'd like to thank everyone for coming out today. Even though we had a little bit of time issues with everything. So this concludes the cellular and molecular biology division. And so we will be taking a picture for everyone involved in this division for social media. So if you'd like to turn on your camera and be inside the thing, you may go ahead and do so now. All right, thank you. If the judges would like to stay behind and deliberate and discuss any comments, all the presenters are free to leave at this point. All right, thank you, everyone.
Good job, everyone.